Okay. Gonna share screen. Okay. So let's go to cardio 1. Okay. So we'll just wait a few seconds, let this load. Hopefully you can see this and it's recording. Looks like it is. As soon as it 30 more seconds. Okay. All right. So this is the first lecture on cardio. So if you see this on your slide or microscope, so that's red blood cells. So that's fluid connective tissue, remember? So these are erythrocytes or red blood cells. These are platelets. Those are pieces of a cell. They're also called thrombocytes, and there are no white blood cells on here. White blood cells are also known as leukocytes. So remember, red blood cells are erythrocytes, platelets are thrombocytes, white blood cells are leukocytes. All this fluid in between would be plasma. So what's the functions of the cardiovascular system? Transport gases like O2 and CO2, hormones, nutrients, waste. There's some temperature regulation, but we're not going into that. That's physiology. Don't forget pH regulation. There's buffers in your blood to keep your blood in a range of 7.35 to 7.45 on the pH scale. Very important. Burn those numbers into your brain. We'll talk about them more. And there's immune functions, you got white blood cells and complement and antibodies and stuff floating around in there, and we'll get to some of that. All right, components, you got the fluid, the pump, and the pipes. So the blood's the fluid, the heart's the pump, and the vessels are the pipes. So the fluid carries the gases, nutrients, hormones, all of that stuff. The heart's the pump, it's 4 chambers. Got atria on the superior or top, and ventricles on the bottom, inferior. The vessels, you got arteries, veins, arterioles, venules, capillaries. We'll get to all of those, but that's the pipes. Okay. So the function of the blood is, like I said, for transport. It's also protection against microorganisms. You got your, white blood cells and your immune system components in there. Also, it keeps you from getting dehydrated. Regulates vital fluid balances, your pH balance in your tissues and your body temperature. So blood is approximately 45% formed elements. And by formed elements, I mean white blood cells, red blood cells, platelets. The rest of it, 55%, is plasma. Stuff's dissolved in the plasma. We'll get to that. Burn this range into your head, 7.35 to 7.45. If it goes below that, it's getting too acidic. If it goes above that, it's getting too basic. Okay, we're not going to worry about the temperature thing. We're not going to worry about this either. Males generally have 5 to 6 liters of blood. Females have 4 to 5. That's more of a body size thing than a male-female thing, though. Sorry. So, 55% volume is plasma. I'm not going to hit you with 92% of plasma is water. 7% is dissolved substances. But yeah, you got proteins, waste, nutrients, hormones, stuff's dissolved in there. But importantly, you have some plasma proteins dissolved in there. And here's 3. Most plasma proteins are made in the liver, but not all of them. Let's do the easy one first, fibrinogen. Think clotting when you see that word. That's the inactive form. It's activated to fibrin, and that helps you make blood clots. Globulins, I want you to think antibodies when you see that word. And albumins, I want you to think help you maintain the viscosity or how thick blood is, and then the osmolarity of the blood. 45% of the blood is approximately cellular. So red blood cells, white blood cells, little platelets, that's your formed elements. And this is just what we said. So fibrinogen, I could do a matching on this. So if I said fibrinogen, and then I put aids in blood clotting, you would match those two up. Globulins, think antibodies, help tag or, help your immune system get rid of foreign proteins. So antibodies would match up with globulin. And then albumin, think maintain osmotic pressure and viscosity of the blood. Now, if you said forms lipoproteins for transport of proteins, fats, and steroids, I can't count you wrong. That's true too, but this is not physiology. Just stick with the easy ones for now. Same thing, globulin, and you could go transport protein, binds insoluble ions, hormones, blah, blah, blah. But we're not getting into all that. Just mainly think antibodies when you see globulin, mainly think osmotic pressure and viscosity of blood when you see albumin, think blood clotting when you see fibrinogen. Erythrocytes, that's got your hemoglobin in there, and it helps you transport the O2 and CO2. Leukocytes, those are your white blood cells, and platelets, that's for clotting. So remember this little saying, never let monkeys eat bananas. So that's a little mnemonic that gives you the first letter of each of your different types of white blood cells. N stands for neutrophil, L stands for lymphocyte, M for the monocyte, E for the eosinophil, B for the basophil. So never let monkeys eat bananas. The good thing about that little saying, it ranks them in prevalence also. So you have more neutrophils than you have lymphocytes. You got more lymphocytes than you do monocytes. You have more monocytes than you do eosinophils. And you have more eosinophils than you do basophils. Now, that's in a healthy person. Now, if something's out of whack, it can, it might mean something's up, like an infection or a cancer or whatever. But as a general rule, that's the prevalence. That's the oral prevalence. So, let's see if we got anything about that. Not on that side. We'll come back to that. So, hemopoesis, that's just making blood. So before you're born, it's being made in your red bone marrow, your liver, your spleen, your thymus. After you're born, it's mainly red bone marrow, mainly in your ribs, your sternum, and your hips, your iliac crest. There's some yellow bone marrow that's mixed in with that, and you can utilize that in an extreme situation. Hemocytoblast is just the stem cell that can become any of these formed elements. So your book calls it something different now. Instead of hemocytoblast, they call it pluripotent stem cell. Same thing. I like hemocytoblast, easier. Hemopoiesis, that's just blood cell formation. So the hemocytoblast can become any of these things. So it can become white blood cells, can become red blood cells, can become platelets. Of the white blood cells, I'm just showing you 3 here instead of 5, because these are the 3 most common: neutrophil, lymphocyte, and monocyte. Neutrophil looks like it has more than one nucleus, but it doesn't. So if you look right there, it is connected. So sometimes you'll see 3 or 4 little lobular areas in here for the nucleus, but it's just one. The lymphocyte looks like it's almost all nucleus, but it's just got a little rim of cytoplasm around it. Almost looks like an eclipse when the moon goes in front of the sun. And then a monocyte that can become a macrophage. So that's a very big one. So look how big that is compared to your red blood cells, and it's not even that big a one. But a lot of times, if you catch it at the right angle, the nucleus will look like a kidney shape. See that kidney shape? And so that's the way you tell those. One thing I forgot to mention. You probably heard these terms before: granulocytes versus agranulocytes. Granulocytes just means they have granules, in the cytoplasm. Agranulocyte, A means without, there are no granules. And don't even worry about what the little granules are, but see all those little dots the artist drew there, and there? Those are granules. And they help them do their job, it's little vesicular packets of stuff. So I never could remember which ones were granulocytes and which ones were agranulocytes. I kept getting them mixed up. And then it occurred to me one day, it has nothing to do with this. It's just an easy way to remember it. Neutrophil, eosinophil, and basophil, those are filled with granules. I know it's spelled wrong and it means something different, but that just helped me remember it. Those are filled with granules. So those are granulocytes. The other two, there's not a granule in sight. I know it's a stupid little saying, but it helps me. So there's no granules in a monocyte and a lymphocyte. Now, don't apply this saying to leukocyte because leukocyte is just all of them, so it wouldn't apply. Leukocyte encompasses all 5 of these. Okay, so this is just showing you that a hemocytoblast can become all of this. There's my red blood cells, there's my platelets, there's my basophil, eosinophil, neutrophil, monocyte, lymphocyte. Go upstream one from the platelets, and you got this big guy here. It's called a megakaryocyte. Sometimes megakaryoblast, they call it. I don't care, either one. But little pieces break off of it, and those become your platelets. So true or false? Platelets are cells. False. They're pieces of a cell. Then I could ask, what's the immediate precursor to a platelet? And you go, megakaryocyte. What's the precursor to all the formed elements? The original precursor, hemocytoblast. Or you could say pluripotent stem cell. True or false? Red blood cells, the mature ones, have a nucleus. False. See, it gets gone pretty early on. And that just leaves more room for the hemoglobin on the inside. So these guys don't live that long. They live about 120 days. Okay, they're not that big. They're about 8 microns across, where a capillary, maybe 10 microns across. They can even be smaller. These things have to line up single file and go through them. And they can kind of bend, so they can even get through areas that are smaller diameter than they are, actually. But I think most capillaries are around 10 microns or so. So these guys have to line up single file to go through them, and that slows them down and it gives it time for gas exchange to occur. So the hemoglobin is the quaternary molecule. It's got 4 subunits basically, and each one of those 4 has an iron in the middle. And so the hemoglobin binds reversibly with the iron atom in there combines reversibly with the oxygen. So it wouldn't be any good to be irreversible, right? Because say when you breathe in, your oxygen is jumping onto your hemoglobin in your lungs, in the capillaries in your lung. Then that blood gets transported, say, to your feet. It's different levels of pH and oxygen levels down there. So it unloads. And goes into the tissues where it's needed. Well, what are those cells in the tissues making as a byproduct? Carbon dioxide. So carbon dioxide can load onto the hemoglobin in a different spot, but it's on it, and then get carried back to the lungs, and then the carbon dioxide leaves and gets exhaled, and then with your inhalation comes in the oxygen, and then the oxygen can load onto the hemoglobin, and then it can do this trip again. So it's a reversible binding of the oxygen. What almost binds irreversibly? Carbon monoxide. Very dangerous. Can't see it, can't smell it, can't taste it. And it doesn't want to let go, so then you can't really carry oxygen. So that's very, very dangerous. So make sure you're if you have smoke alarms, make sure they have a carbon monoxide detector on them too, because that can be just as dangerous as smoke, or even more, because you can't smell it. So yeah, just check your smoke alarm. Most of the new ones have it now. All right, blood types. So we talked a little bit about antibodies and antigens before. Normally you'd think of an antigen as something foreign, like a bacteria or a virus or something like that. But you can have self-antigens too. Antigens are usually made of protein. And so let's look at these red blood cells. So here's our type A, B, and O. You probably heard of those. So we're not even talking about positives or negatives like O positive and O negative. That's a different thing as far as, you know, a protein or an antigen on there. You'll get that in physiology. We're just sticking with the basics. This is anatomy. So type O, if you look on the surface, there's no A antigens and there's no B antigens on there. Okay? Where if you look at type A blood, it has A antigens on it. You look at B blood, it has B antigens on the surface. If somebody's type AB, they have A and B antigens on the surface. Okay. Well, here's what's weird, and nobody really understands how this happens. But if you're type O blood, you have none of these proteins on your cell surface of your red blood cells, none of those A's or B's. But you're making antibodies to A and antibodies to B, you know, to the antigens. It's no problem because this is floating around in your plasma, but there's nothing for it to stick to. Remember, they stick to things. And because there's no antigens on your red blood cells. So that's cool, right? So this blood cell with no antigens on it is the universal donor. I have asked that before. So when you donate blood to somebody and they take your blood and your plasma comes out in there too, but they pour off all of that plasma because that's got the antibodies in it. So they're probably going to get rid of that. And then they're going to wash these guys off and then resuspend these red blood cells in a saline type solution. So now if you're type O or whatever, and you're giving somebody your blood, you're only giving them your red blood cells as a general rule. So that's why this guy's a universal donor. There's nothing that's going to stick to him, right? So he can donate to type A, type B, type AB. Doesn't matter what's in the plasma. It doesn't matter if they got antibodies because there's no antigens to get, to stick to. For your antibodies to stick to. Now, your type AB blood, this is considered the universal recipient because look in their plasma, they're not making A or B antibodies. I don't call them anti-A and anti-B, I just call A or B antibodies. So it doesn't matter what you put in there. You can put A blood or B blood in here. No, it's cool because this person in their own plasma has no antibodies, so nothing is going to stick. Now, type A, look at this guy. He's got A antigens. Well, he better not have A antibodies or it would stick and clump, right? But they're making B antibodies, which is cool because there's nothing for the B's to stick to. Where look at type B, has surface, has B antigens on the surface, right? But there are no B antibodies to stick to them. It has A, this person's automatically making A antibodies, but that's cool because nothing to stick to. So in other words, if you're type A blood, you have A antigens and you have B antibodies in your plasma. If you're type B blood, you have B antigens on your cell surface and you have A antibodies in your blood. And then if you're AB, you have both types antigens on your surface. And of course, you have no antibodies in your blood. Type O, no antigens on the surface. And you have A and B antibodies. So I could ask you, what's a universal donor? You go, O. What's a universal recipient? AB. Now, sometimes I ask a little thought question. Somebody comes into your emergency room and you have no idea what blood type they are. They were in a car wreck, they're knocked out, and you need to give them a transfusion. They need some blood. So you can take a drop of their blood and you put in a little well, and then you drop some, say you drop some A antibodies onto the blood that's in that well. And if it clumps or coagulates, then they're either type A or they could be type AB, because those A antibodies are sticking to either A antigens on type A blood or A antigens on type AB blood. So you've got it narrowed down. So next you would need a fresh drop of blood in a well, and you would drop some B antibodies on it. Well, if those stuck, then you would know they're type AB. If they didn't stick, then you know they're only type A. So it's like a little thought question. I have asked a question like that before. All right, there's sickle cell. Sickle cell can cause your red blood cells to sickle. Hence the name sickle cell. And you probably heard of sickle cell trait versus the full-blown sickle cell, so you can be heterozygous or homozygous for this problem. Heterozygous means you have one bad copy on your allele. You have one allele that's bad with the DNA, and then your other copy is fine. So that's heterozygous. So that's called sickle cell trait. Where if both copies are bad, then they're way more prone to having a crisis like this. And you can see that this is not going to go through the capillaries as well. And so they can get clogged up. Now, how does something like this get preserved down through history? Well, it could have a benefit if you just have the trait, because it can make you less susceptible to malaria, they think. So I have asked that before. Sickle cell trait can help protect you from what disease? Possibly malaria. And so that's how that has gotten passed down through the ages, is because it could provide some protection as long as you're not homozygous, right? Okay. We talked about the never let monkeys eat bananas. There we did. I'm not going to ask you 70% versus how. I'm not going to ask you what percentage they are. Just know you got them. Neutrophils are highly mobile phagocytes. I go, what's a neutrophil's job? Highly mobile phagocyte. What's an eosinophil's job? Well, certain parasitic infections they can help fight. They can go up in certain allergic reactions too. But generally, if you go into the doc and your eosinophils are way high, they're going to probably check you for some type of parasite. Basophil can release histamine. And histamine, so like you get bitten by a mosquito, and that little whelp you get, that's a local histamine release, or a bee. Bee stings you, you get a whelp. That's a local histamine release. But if you're allergic to that bee, it's possible you could have a systemic histamine release in various other places in your body. And that can be very dangerous. That can lead to what they call anaphylactic shock. And so it's like an asthma attack, and you can die from that. So they might have to give you an antihistamine, such as maybe Benadryl, to help, stop that. And so that can help stop that. And if it's really bad, they might even have to give you like an epinephrine or something, like that, that's going to dilate your bronchioles so that you can breathe better. So that's in a real severe reaction. Lymphocytes, mainly I want you to think antibodies. Yeah, there's other there's B cells make the antibodies. T cells, they do a lot of other things. And there's more subdivisions of these cells than I'm giving you. And we'll talk about them more when we get to lymphatics. But right now, just remember B cells make antibodies. Monocytes can become macrophages, and those are highly mobile phagocytes too. Platelets we talked about, they come from a megakaryoblast or megakaryocyte. They're little pieces. They help with blood clotting. So the heart, let's just do the pieces and parts. You got atria on the top, ventricles on the bottom. A pulmonary circuit is just where it's pumping from the heart to the lungs and back. And the systemic circuit is where it's going from the heart to the body and back. So this is the heart. You're looking at the front of it. There's a big old aorta. There's a superior vena cava vein that's bringing blood in. And we'll go into that a little more in depth in a little bit. So 3 layers of the heart. So if you look at the inside, see, this is the inside of a ventricle. That's the endocardium. The muscle wall would be the myocardium, and then the shiny outside part of the heart would be the epicardium, or you might remember visceral pericardium from your first test. So that's visceral pericardium, but the heart's kind of in a sac. So there's another layer outside of that that's kind of loose, and you can cut through it. That's the parietal pericardium. So if you were to open up a cat, and you look, you go, well, I see the heart, but I can't really see it. It's almost like it's in a little bag. That's that parietal pericardium that you're seeing. You have to cut through that with scissors, and then you see the nice shiny heart underneath, and you'd be looking at the visceral pericardium or the epicardium, which would be the shiny layer. So in other words, the shiny layer right here, that's your epicardium. Okay. Let's look at this. Hold on. That would be your myocardium. That would be your epicardium. And the shiny stuff on the inside of your chambers would be your endocardium. Okay, we'll come back to that. Okay. So the fibrous skeleton, I don't go into that too much, but if you dissolved everything out of the heart except the connective tissue, it would leave kind of like a collagen matrix in the shape of a heart. So that gives a place for the valves to attach. It supports the muscle cells, helps distribute the force of contraction. It keeps the heart from overfilling because, it's got to keep its shape, because you don't want to overfill because you'd stretch those sarcomeres apart. And it physically, but also electrically isolates the atria from the ventricles, right? So it's almost like insulation in between your atria and ventricles, because you don't want those to depolarize at the same time. And we'll talk about that on a lecture 2, I think. So you want the atria, all the cells in the atria to depolarize so that they can contract. And then you want all the cells in your ventricles to depolarize so that they can contract later. You don't want these two things to happen at the same time. So there's like a little insulation to avoid that depolarization from spreading throughout the whole heart simultaneously. All right, so it's similar to skeletal muscle because it's striated. The heart cells can contract without signaling from the nervous system. So if you took a little frog heart and put it in a dish, it would continue to beat for quite a while, because these cells are autorhythmic. The muscle cells are connected by a little intercalated disc. We talked about that on test 1. And there's little gap junctions in the intercalated disc that help spread that signal for depolarization out. Okay, so the intercalated disc hold the cardiac muscle cells together, right? And the gap junctions that are in those intercalated discs allow ions to pass from one cell to the other. So that's that depolarization I was talking about. So they have to work together, like the atria have to all contract at the same time, all those cells, and then the ventricular cells all have to contract at the same time. So this is just showing you pulmonary circuit versus systemic circuit. They didn't draw all the blood vessels in the arms and the leg. I guess they just saving space here. But look at the right side of the heart. So here comes blood from the body in from the superior and inferior vena cava. It's going in the right atrium, right ventricle. Then it's going to the lungs to get oxygenated. Now it's coming back from the lungs, but it's going to the left side of the heart. See? And that's why they drew the left side red, because it's oxygenated. So that oxygenated goes to the body. This deoxygenated blood goes to the lungs to get oxygenated. So the pulmonary circuit is just from the heart to the lungs and back. The systemic circuit is from the heart, well, in this case, the left side of the heart, to the body and then back. So that's the difference between the pulmonary versus the systemic circuit. Who has to work harder, the right ventricle or the left ventricle? Well, the left ventricle has to work harder because it's pumping to the body, and you have more. It takes more pressure. The right ventricle just has to pump to the lungs. They're pumping, so whatever blood the right ventricle pumps, say it's 70 milliliters, maybe 100, the left ventricle is going to pump the same amount, but different pressures. So, true or false? The right ventricle pumps more blood than the left ventricle. False. It's a closed system. They both have to pump the same amount, but different pressures. That's why that left ventricle wall is thick. And I'll show you that in a little bit because it has to be strong. Okay. So the atria just pump to the, their walls are not very thick because they don't have to pump very far. They just pump to the ventricles. So we'll talk about SA node and AV node on another lecture. That's the that sets the pace or the rhythm of your heartbeat. So those they depolarize at a certain rate. We'll get to that. Superior and inferior vena cava we talked about. Then you got coronary arteries and veins that's on or in the heart itself. And don't worry about the foramen ovale. We'll get to that later. But let's just learn some parts. This is a valve in between the atrium and the ventricle on the right side. So I would call it a right AV valve. This is a valve in between the atrium and a ventricle on the left side. So it's called the left AV valve. I just call them left and right AV valve, and that way you never go wrong. Okay, and because they do have other words, and we'll do it on another lecture, tricuspid and bicuspid valve. And then they also have one of these valves they also call the mitral valve. So for now, let's just call them right AV and left AV. That way you never go wrong. Look at the left ventricle wall, thick, very strong. Look at the right ventricle wall, not that thick. Don't look at, that's the interventricular septum. It's always thick, right? This is the apex. So sometimes I just stick a pin in there on a sheep heart. What is that? Apex. What is that layer? Myocardium. What is that layer? Epicardium. What is that layer? Endocardium. What is this chamber? Left ventricle. What is this chamber? Right ventricle. What is that chamber? Right atrium, left atrium. Got it? Pulmonary circuit, systemic circuit, we talked about. And then this is just showing you a neutrophil, a lymphocyte, and a monocyte, because those are the three most common, especially neutrophils and lymphocyte. There's platelets, pieces of them make a karyocyte. There's my intercalated disc. That's just in between heart muscle cells. And then it's got little gap junctions in there. Can't see them here, but that passes ions so that they all are, connected as far as they all will depolarize at the same time. It's got little stripes in them. It's got striations, but you can't see them that well here. This are horrible horrible slides, but this is an artery, that's a vein. Arteries tend to have a thicker muscle layer around them. That's called the tunica media. I would show you a better artery than this. It's going to look like a circle, and it's going to have really thick muscle right here. So the tunica interna is simple squamous right here with some elastic fibers, under it. Tunica media is the muscle. Tunica externa, that's the connective tissue on the outside. This has the same three, but it's not as thick, right? Especially the media. The old names, tunica intima, tunica media has always been tunica media, and they call that tunica adventitia. Don't use that here, but remember that word because we do use it around the esophagus on test 6. There's a tunica adventitia around the esophagus. For us, just go tunica interna, tunica media, tunica externa. And so what vessel is this? You go artery. What's the pointer on? Tunica media, that kind of thing. Another thing on an artery, see the way it's ruffled? That's because it has a lot of elastic fibers mixed in here with the arteries. The veins, this is not a good example. They're going to be more smooth because they're not, basically it's not as much as far as elastic fibers. Then we're done. Okay, I'm going to stop the share and then we'll post this. Let's see, stop share. And then end meeting. End meeting for all.